Hello, students. Very good evening to all of you. So I hope others have joined in the YouTube as well as they're going to join in the Zoom too. So before we start the session, I'm going to launch a technical check. So please try to uh, answer this question so that I would know if my audio and video as well as the board is visible. Yes. So let's start today's session. So we are going to have a very quick glance about cell biology. So where we'll be studying about cell structure and function. So give me a second. Good afternoon. Yeah, good afternoon, guys. So before we start this session, so I'm going to tell you that it's not going to be a very detailed session. So we are going to have a glimpse on this chapter, and we are going to highlight some of the things that you have already learned. Okay. So basically, enable that we are starting for 11th standard, we are going to concentrate on the term 2 syllabus because it has high weightage chapters. So anyways, we'll start today's session. So before we go to cell structure and function, okay, so give me a second. Yeah, before we go to cell structure and function, so let us start by answering few questions. Okay, so here is your first question, students. Select the wrong statement. Okay, so let us see what you have learned in your past classes. The first option is bacterial cell wall is made up of peptidoglycan. Pili and fimbriae are mainly involved in mobility of bacterial cells. Cyanobacteria lack flagellated cells. Mycoplasma is a wall-less microorganisms. So I will launch an MCQ poll for you so you can answer this question. Okay. Give me a second. So here's going to be your MCQ poll. So you have a minute to answer this question. So please try to answer this. Select the wrong statement. Yes, bacterial cell wall is made up of peptide. Look here, guys. Wrong statement. It says, please read the question carefully. Okay. Bacterial cell wall is made up of peptidoglycan. Pili and fimbriae are mainly involved in mobility of bacterial cells. Cyanobacteria lack flagellated cells. Mycoplasma is a wall-less microorganism. So this was 2016 NEET question. So let's see. So one of you have answered, and it's already almost 50 seconds up. Okay. All right. So we're almost reaching to our 1 minute. So I will end the poll right now. So one of you have answered option A. Okay. So let us see one option after another and try to analyze our answers. All right. So here we have bacterial cell wall is made up of peptidoglycan. This is an absolutely right answer. Okay. Peptidoglycan is also called as murin. Okay. Give me a second. Okay. So we have murin. Okay. So murin layer. It is also called as murin layer. Whereas the second option, okay, if people would have thought second option is the right one, okay, pili and fimbriae are mainly involved in motility of bacterial cells. Okay, so pili and fimbriae, okay, so these are the structures that arise from the cell wall of the bacteria. So they are involved in motility. Okay, they're also involved in attachment. Okay, if they want to attach, why is this? Okay, give me a second, people. Okay, so they are also involved in attachment. Okay, so they're also involved in sex reproduction. Okay, so or sexual reproduction. So motility, attachment, and sexual reproduction are one of the main functions of pili and fimbriae. Okay, we're going to learn about them, no problem. So next one, cyanobacteria lack flagellated cells. Okay, so we'll come to this option. Give it a second. Next one, mycoplasma is a wall-less microorganism. So mycoplasma is one of the smallest living cell known to mankind, right? So they don't have a cell wall. So this is a right option too. Whereas when it comes to cyanobacteria, okay, cyanobacteria are both motile and non-motile. That means they have, okay, I will write it here. They are both motile and non-motile. All right. So when they are both motile and non-motile, okay, that means this option is particularly wrong, right? So your option should be C. I hope you have understood, okay, why option C is wrong. Okay, so since they have, okay, they don't have a cell wall, they have been specially placed under a different group itself. Okay, so we will discuss in detail as we go ahead with the classes. Okay, so let's see what we have in the next slide. So here you have a match the following. It's going to be interesting. You have column 1 and column 2. So here you have 4 options. For the first option is thylakoids. Second option is cisternae. Second option is cristae. Third option is cisternae. Fourth option is chromatin. And then you have the following options in the column 2. So try to match these in a right fashion. I will give you 2 minutes for this match the following. Okay, since it's a beginning. Okay, hold on. Yeah. So let's see, what do we have? So the poll is launched. You have 2 minutes to answer this question. Try to match the right options, people. Try to work out in your sheets. So we have column 2 and we have specific options given here. Okay, we have I, that is the first option in Romans, that is dish-shaped sac in Golgi apparatus. Okay, and the second one is condensed structure of DNA. And the third one is flat membranous sacs in stroma. Okay, and the fourth option we have is, okay, infolding in mitochondria. Excellent. Okay, within 1 minute, we have 2 people answering, option C. So I'll end the poll. So we here. So let's see if you are right. So I will just mark option C. Okay, I'll just hold on. Yeah, I'll just mark option C because you both have answered option C. So you're telling thylakoids are for, that is infolding in mitochondria. Okay, so what are thylakoids, people? So when we talk about plastids, right? So when we talk about plastids specifically, okay, plastids, and we talk about chloroplast, right? When we talk about chloroplast, right, we talk about very important regions of your chloroplast, that is grana and stroma, right? So in grana region, you have, okay, in grana region, right? So you have this pile of coin-like structures, okay, which we call it as thylakoids, okay, that the disc-like structures called as thylakoids. So these thylakoids are basically present where it is present as flat membranous sacs in the stroma of what? Of chlorophyll, okay, chlorophyll. So they are responsible for performing light reactions, that is harvesting light, okay, for photosynthesis. So this is about thylakoids. And next one, what is cristae? Okay, so what is cristae? So usually what people will confuse with cristae and cisternae, they will try to confuse with it. Okay, cristae are basically found in mitochondria. So what happens, we have, let us say that we have, okay, a shape of mitochondria like this. Now what happens, the inner membrane, so mitochondria is a double membrane organelle. So the inner membrane of the mitochondria, what it does is it will start folding inwards and outward, just like this. It will start folding inward, okay? Dividing the mitochondria into two essential compartments, okay? So these things we call it as infolding in mitochondria. Basically, we call this infolding as cristae, okay? Next one, cisternae. All right. So what is cisternae? Usually when we talk about Golgi apparatus, okay, in Golgi apparatus, we have highly convoluted foldings, right? Something, sorry. I'll just write it like this. Okay, something like this. Okay. So these foldings are present in Golgi apparatus. So they create this concave and convex, okay, portions of your Golgi, which are called as cis phase and trans phase, where it is called as, okay, entry phase, okay, entry phase, or we can also call as the other phase as maturation phase, okay. Anyways, you need not worry about what I'm talking about now, like entry phase or maturation phase. So basically what's happening is, so these cisternae are these folds that are present in Golgi apparatus, okay, where what happens when the molecules, so like biomolecules, like proteins, lipids, okay, or any of this like steroids that has to be modified or transported, okay, they enter through one of the phases of, okay, one of the phases of cisternae and they exit through another phase, another phase of cisternae. So the maturation process will happen here. All right. So we know that the cristae is present in mitochondria and the cisternae is present in Golgi apparatus. And our final option that we are left with is chromatin. Okay. So what is chromatin fibers? So basically, when I ask you a question about, okay, the size of the DNA that we have, right? So the size of the DNA that we have is very tremendous, right? So if you just try to unwind the DNA that is present in the one single cell of human beings, you can stretch from Earth to Moon and Moon to Earth. Okay. So that is the length of DNA that is present in every single cell in human body. And roughly human beings have around 30 trillion cells. Okay. Why I'm telling this is because all this information that is present in DNA is very, very, very vital. Okay. Very, very important. So we cannot miss it when we transfer the information to the next generation cells, like when the cell division happens. So now what happens, our cells have evolved a way to pack this DNA. So when they condense this DNA, they try to pack this into thick fibers, and these fibers, we call it as chromatin fibers. Okay, so we have here, see, condensed structure of DNA. All right, people. So I have told you what are the right options. Now tell me, I've told you what is the right answers. Now, can you tell me which is the right option right now? Because we have to check it now. Can you tell me? Now I'll relaunch the poll. You have to tell me, okay? Please tell me which is the right option. So you might have noted the answer. I will understand if you're following me. Which should be the right answer? D. Yes. A, thylakoids are flat membranous sacs, correct? And then we have B, 4, right? That is B, cristae, that is infolding of mitochondria. C, that is cisternae, disc-shaped sacs in Golgi apparatus. And then we have D, that is chromatin, condensed structure, present in of DNA. Excellent. Thank you for following this class. Okay, now it makes it more interesting. So fine, since we have concluded with, a prior knowledge of our topics, so let's see what we have with the topic itself. So we are going to have an introduction about cell, and then we will study some vital concepts about what is the cell structure and how it contributes to the function. Okay, so basically all living organisms are made up of cells. Okay, whether you take a prokaryotic organism, okay, or unicellular eukaryotic organisms, okay, or you take tissue level organisms or organ level organisms or organ system level organisms, intelligent, full, bigger, smaller, okay, all the living organisms are made up of cells. So every individual cell that is present in every single organisms is capable of performing all the living functions, like how we do. We respire, cells respire, we grow, cells grow, we transport our nutrients, cells transport our nutrients, we communicate, cells communicate, we respond, cells also respond. Okay, we divide, cells also will divide. Okay, each and every single cell is capable of performing and living independently like every single organism. Okay, so they combine together to form one coordinated function, okay, to result in tissues, organs, organ systems, and complex organisms. So all the living organisms, their characters, the way they express, okay, their capabilities, okay, or the food they eat, everything depends upon the cell itself, whether it is plants, bacteria, or humans, or any other animal on this planet. So that is why we call cell as a fundamental, structural, and a functional unit of all living organisms, right? So they make up life. All right, so let us see what happened with the discovery of cell. So initially, back in the days, when we look back, so there was very minimalistic idea about what is cell. Okay, people did not predict the existence of cell, or that they didn't talk about the cells until the development of this glass, like what do you call, magnification glass by Leeuwenhoek. Okay, he was a very good spectacle maker. So he started developing this hand lenses, and finally he started developing more, we can talk about around 50x or 45x kind of low magnification microscopes. So as it advanced with the magnification and microscopy, okay, primitive microscopy, so the first person to even tell that every living organism, okay, all the plants that I have observed, okay, is made up of this comb like structures, okay, like this honeycomb like structures, and they are all looking like, they're like small units which combine together to form a complex unit. So this Robert Hooke, okay, called it, he just told that, okay, this is called as a cell. Each honeycomb like structure is a cell, and several honeycomb like structure combine together to form a complex organism. That's what he said. And later what happens, once the Robert Hooke gave the idea of cell, Leeuwenhoek developed more and more interest in studying microscopy. So he started analyzing all the samples, for example, the scrapings of the teeth, the fecal matter, the blood samples, okay, the contaminated water, the food, okay, whatever, wherever he can get his hands on. So Leeuwenhoek was highly motivated to understand what is below this microscope. Okay, so once this discovery of this living organisms which are invisible to our eyes, which is called as microbe, okay, an entire new world opened up to biology, where we have been studying the characteristics of the cells, characteristics of diseases, characteristics of so many different organisms. Okay, and it's still happening, and it is going to happen, and it has become a never-ending subject. Okay, so this is a brief history of how the discovery of cell took place, and how Anton von Leeuwenhoek was successful in examining the living cells, okay, analyzing various samples that was around him. Okay, so as the discovery of cells took place, now people wanted to credit and understand what is the real characteristic of a cell. So how important is the cell? So as this curiosity grow, and then we have two important scientists to contribute, okay, to give a very foundation for, okay, to give a appropriate value for the cell, where they proposed cell theory. Okay, they called something called as cell theory. So these scientists are called as Sheldon and Schwann. Okay, so Sheldon and Schwann are two independent scientists, where you have Sheldon, who observed plant cells, and he tried to understand all the plants were composed of cells. Okay, so he was a botanist. Whereas Theodor Schwann, he reported so many different types of organisms. He studied both plant cells as well as animal cells. Okay, he tried to understand what is plasma membrane. Okay, he tried to understand that there is a protective lining, there is a semi-permeable membrane that is present. Okay, so which he called it as a plasma membrane. Okay, which he called it as he didn't call it as a plasma membrane. He discovered plasma membrane, but he could not explain it. Okay, and he discovered the same thing in plant cells too. So finally, he come to both of them come to this hypothesis, telling that all these plants or animals, okay, we have so much difference between plants and animals, yet irrespective of the differences, okay, what we can observe them is All of them are made up of cells and the products of cells. So primitively, when the cell theory was suggested, right, when the cell theory was told to the world, they just told this thing. What did they tell? Sheldon and Schwann told that animals and plants are composed of cells and the products of cells. Okay, so this was the, what do you tell? This was a statement given by these two scientists. And Virchow, okay, Rudolf Virchow later expanded this hypothesis, okay, where he tried to understand the significance of cell division. So as the cell divides, he gave a very wonderful statement that for every new cells that has to come to existence, okay, it has to come from the pre-existent cells. That means one mother cell has to be there so that a daughter cell can be born. So this is the first statement, okay, or modified statement of cell theory, okay, Rudolf Virchow gives, okay, and he calls it as, okay, omnis cellulae e cellula, okay, that means all cells exist from pre-existing cells, okay. And finally, after his statement, now the cell theory was completely modified, okay, not completely modified, it was modified for the modern understanding, where we have two important statements. So cell theory states that all living organisms are composed of cells and the products of cells, so which is given by Sheldon and Schwann. And next, Virchow's contribution is all cells arise from pre-existing cells, okay. So this is the statements given by these three scientists combined together. And then finally we have the cell theory. Now it might be a generic statement telling that, okay, all living organisms arise from pre-existing cells, or we can talk about all living organisms are, composed of cells and their byproducts, okay. But if you understand it even in a deeper sense, you'll see that this statement summarizes the whole of your microbiology, right? So that is why we call these two statements as the pillars of cell biology, where we call it as cell theory. Fine. So coming to understanding the cells, first we have to go for the primitive cells, that is prokaryotic cells. Okay, so when the life originated on this planet, so the very first organisms, like very organized cells, what we call it as, so those organized cells were not as advanced as the eukaryotic cells. Eukaryotic cells function and the way they perform is very complex compared to that of prokaryotes. Okay, so to just split the word called as pro means early, okay, or we can call it as old, okay, or we can call it as less advanced, okay, just like your iPhone. If I have an iPhone 8 or iPhone 7, okay, and if you have an iPhone 14, so which is more advanced? iPhone 14 is more advanced, right? New features and new software and new accessibilities and all that. So all these prior ones that we had before, we call it as early phones or early things, right? Similarly, even in cells, there is some advancement. So these early cells to be evolved, we call it as prokaryotic. Pro means early, karyon means nucleus, okay? So the organisms with early or less advanced nucleus are called as prokaryotic cells, okay? So coming to the cell organelles of a prokaryotic cell. So what do we find as a prokaryotic cell? So what do we find in prokaryotic cell is almost same as an eukaryotic cell, but there are some significant differences in prokaryotic cell structure. What are those? Let us see, let us understand. So if you see the outermost layer of your prokaryotic cell, it is covered by a capsule, okay? So, or a glycocalyx layer, okay? So, or a mucilage we call it as. So this will shield this bacteria from so many different things, okay? From the entry of pathogens you might understand, or trying to infect the cells, okay? Or trying to, recognize their own strains, okay? So this is about the capsule. So below the capsule, we have cell wall, and the cell wall with the prokaryotic organisms is very, very unique, okay? So it is made up of peptides and glucose, which we call it as, we'll talk about it. So which is peptidoglycan layers or murin layers. So since most of the prokaryotic, all of the prokaryotic organisms are bacterium, okay, or bacteria. So usually based on the characteristics of the cell wall, we can categorize this bacteria as gram positive and gram negative bacteria, okay? So I hope you have heard about gram staining. If not, we will still talk about it. So this is a revolutionary discovery of understanding the differences in bacteria. Okay. So now, for example, if you talk about gram positive bacteria, all these gram positive bacteria are good bacteria, and gram negative bacteria are known to cause diseases to mankind. Okay, this is one simple way to understand gram positive and gram negative bacteria. So it become very easy for people to understand what type of bacteria is infecting, or what type of bacteria is present in certain foods or certain environment, etc., etc. So below the cell wall, which is made up of peptidoglycan, as I said, is your plasma membrane. And the plasma membrane, like that of the eukaryotic cell, the plasma membrane is again made up of peptidoglycan. It is made up of phospholipids. It is made up of phospholipids, okay, phospholipids and proteins. Okay, so it is a double membrane system, okay, like that of your eukaryotes too. Like how we have eukaryotic plasma membrane, like you have a double membrane system. Okay, so you have phospholipids and then you have proteins, right? We'll talk about the structure of plasma membrane so that we will understand. So they essentially perform similar functions like how the eukaryotic cells perform. So what is the function of plasma membrane? Basically to allow the wanted substances or exit the unwanted substances out of the cell. So that means semi-permeability. So that is the function of plasma membrane. And then they have a cytoplasm. And within the cytoplasm are several cell organelles. For example, they have ribosomes, okay, and they have, okay, nucleus. And nucleus is not real nucleus. We call it as nucleoid. Okay, so the major difference between prokaryotic cell and eukaryotic cell is, if we see the cytoplasm of prokaryotic cell, we can see that all the genetic material. So, okay, this is all the DNA that the bacteria have. So all this genetic material is just nakedly floating in the cytoplasm without any protection. Okay, there is no protection of a nuclear membrane, right? So there is no protection. Since there is no protection, usually, so this particular genetic material that is present in the bacterial cell is called as nucleoid. Okay, so the nucleus region of the bacteria cannot be distinguished, okay, into nucleolus, okay, into nucleolus, and then nuclear plasm or nucleoplasm. So all of them are one, it's all mixed up. Okay, so essentially both of them are performing the same functions, that is preparation of RNAs, okay, and cellular expressions, or etc., etc. So there is no clear distinguish between the roles that has to happen in different regions of the nucleus. All of them are mixed up, and the genetic material is naked. And the material that is naked is usually circular DNA, okay, double-stranded circular DNA that is present within the nuclear region. Okay, and coming to the cytoplasm, they do show the presence of, okay, outside nuclear, okay, outside nucleus, okay, or nuclear genetic material. Okay, so genetic material, and this outside nuclear genetic material is usually in double-stranded, okay, circular DNA, and which we call it as plasmid DNA. Okay, you can see in the slide, okay, which we call it as plasmid DNA. So this is a very important point that you have to remember when it comes to prokaryotes, and even in eukaryotes, some of the unicellular eukaryotes like yeast and your protozoans, there are plasmid, but bacteria are, you know, the first one to show this extra chromosomal DNA, which we call it as, I'll just write it here, okay, which we call it as extra chromosomal DNA. So what is extra chromosomal DNA? Extra chromosomal, we call some genetic material which has the capacity to control certain function, right? So we call nucleus as a brain of a cell, or the control machine of the cell, right? So why do we call nucleus as a control center of the cell? Because nucleus will try to command all the cellular organelles to perform a specific function in an orderly fashion. Okay, so imagine, apart from the nucleus, if there is genetic material, right? If there is genetic material outside the nucleus, and this genetic material outside the nucleus, we call it as extra chromosomal DNA. And in bacteria, we find this double-stranded circular DNA as plasmid DNA. Okay, and we have ribosomes. Okay, ribosomes in the cytoplasm. Usually, the ribosomes that are present in your prokaryotic cells are of 70S type. We'll talk about this, what is S and what is 70S type. Okay, so coming to the external features of this cell, when I look at in the microscope, okay, when I look into the microscope and observe these bacterial cells, what can I find? I can find this long whip-like structures. Whip means, right, like usually we beat the horses or we beat cows or something, okay, when they wanted to run or something like that, or punish people. That is called as whip. Okay, so whip-like structure, which is helping this bacteria to move. Okay, so this motile organ, we call it as motile organ or motile organelle, is called as bacterial flagellum. Okay, the flagellum helps in movement of the bacteria. And apart from the flagellum, they have additional cellular, okay, projections, something like pili. Okay, they show the presence of cilia. Okay, they show the presence of fimbriae, okay, etc., etc. Okay, so it goes on and on like this. So this is a very glimpse of what are the organelles that we can expect to study in a prokaryotic cell. So let us go one by one and try to understand different cellular organelles in prokaryotic cell. So I hope that you are able to understand. If not, please try to let me know. Okay, so fine. So as I said before, right, coming to the cell envelope of a prokaryotic cell, okay, coming to the cell envelope of a prokaryotic cell. So I told you that prokaryotic cell envelope, basically the outermost layer of this prokaryotic cell is called as glycocalyx layer, okay, or we call it as mucilage, okay, mucilage layer, glycocalyx layer or mucilage layer. So the role of this glycocalyx layer or mucilage layer, okay, sometimes the presence of this glycocalyx layer or mucilage layer can help this bacteria to cause a disease. Okay, first thing, it will protect the cell from outside infections by producing certain, okay, proteins or toxins, etc. Okay, so this is a function. It helps in recognition of its own strain. So that is the use of glycocalyx layer. And below the glycocalyx layer, we have a cell wall. Okay, and the cell wall is basically, okay, made up of peptidoglycan. Okay, peptidoglycan. Okay, so basically when we talk about peptidoglycan, we have N-acetyl muramic acid is one of the examples of the type of peptidoglycans that are present in your bacterial cells. Okay, N-acetyl muramic acid is one of the examples of your peptidoglycan that is present in the bacterial cell wall. So this peptidoglycan layer of the bacterial cell wall is also called as a murin layer. Okay, murin layer. So usually they ask you, okay, what is the name of the cell wall that is present in bacteria? Okay, so you might not get an option called peptidoglycan or the contents of this peptidoglycan as N-acetyl muramic acid. Okay, so they might just give you as murin. So your answer is right. The murin is right answer because a common name for peptidoglycan is also called as murin. So below the cell wall, okay, so basically the cell wall composition is made up of peptides, that is proteins, and glycan, that is sugar. So when you talk about your plant cell wall, basically you have cellulose, okay, and hemicellulose. Most of the components of the plant cell walls are, or any cell wall that is made up of cellulose, is minerals, sugars, and very little amount of your proteins. Whereas here you see that there is significant amount of proteins as well as your glucose. Okay, so based on the cell wall thickness and the composition, okay, so now what happens is the cell wall try to respond to the Gram staining. So usually whenever we perform the gram staining, okay, I'll just write it here. Okay, gram staining. So gram staining is a technique where we try to understand different classes of bacteria based on the cell wall composition. So when we talk about gram positive bacteria, so we call certain bacteria as gram positive bacteria because they respond, okay, they respond to gram staining. So what do we do in gram staining? Usually it's a staining technique, like how you use methyl blue, right, safranin, etc. So there is a procedure for conducting a gram staining where we use crystal violet, okay, iodine, and then we use alcohol for removing the excess stain. And then finally, when we observe it under the microscope, the cells will appear either violet or they will appear pink. Okay, if they appear violet, that means these cell wall is thicker and they have significant content of these glycoproteins. So that is why it takes up the stain. So these are called as gram positive bacteria, whereas the cells which appear pink, for example, the bacteria which is present in your curd is your gram positive bacteria, whereas the cells that appear pink are the cells which have very thin cell wall or peptidoglycan layer. They will not be able to take up this gram stain. Okay, so when we add safranin, the cell will turn pink. When they turn pink, so that means they are gram negative bacteria. So most of the disease causing bacteria have belonged to the gram negative class. Okay, so coming to the next layer, next protective layer that the bacteria have is a plasma membrane. And the plasma membrane, as I said, is a semi-permeable membrane, which is almost similar to that of your eukaryotic plasma membrane. It is a double membrane bond system where they have, okay, the first content you have your phospholipids, okay, phospholipids, and then the second contains content, or the highest content you have is proteins. Okay, so the phospholipids are responsible for not allowing any of these water molecules to enter from the cells, whereas the proteins that are present in the plasma membrane are responsible for trafficking things. Okay, so they will be trying to understand or control who gets inside the cell or who goes outside the cell, as simple as that. Okay, so this is about the protective layers that are present in your bacterial cell envelope. Okay, coming to the next important, 1 second, we have a chat. Okay, sir, it's unable to see. Okay, I can't understand this. Unable to see the beam color. Can you just repost your question? Or the ink color, you mean? Or the annotation color? Should I change the annotation color or something like that? Please let me know again. Okay, so yeah, 1 second. I will just address this issue. Yes, ink color or red color you're not able to see. All right, I will just try to change the ink. Give me a second. So so where do we have I'm sorry guys. So basically even I wanted to change the ink, but I don't have an option here to change the ink. Okay, or basically maybe I don't know where is the option. So is it okay that if we can try to adjust in this class and I can send you this PPTs with annotations, if that's okay? Because I'm not able to find the source, or if you have an idea about how to change the ink, we just put it in the chat box so we can just try to, correct it as fast as possible. Okay, so I'll continue on with the class, just put the message in. Okay, so anyways, next important organelle, cellular organelle. Okay, you have to remember this. Okay, because it's very significant for the functioning of a prokaryotic cell. Okay, the two important organelles that we're going to study is mesosomes and other one is chromatophores. Okay, so what are mesosomes? So what happens is you have a continuous lining of this plasma membrane, right? So when I think if I write it on the black background, it should be visible. So if this is your bacterial cell So usually plasma membrane is continuous with the cell wall and it envelops the entire cell, right? So at certain points, what happens, you can see here, you can see that there is a inner folding, okay? You can see that there are inner foldings that appear within the plasma membrane. So just because there is an inner folding, what is the significance of this? The significance of these inner foldings in the plasma membrane, okay, will help this bacteria, okay, in the process of respiration, okay, in the process of respiration. They help in the process of respiration. So how do they help in the process of respiration? It's a very, very important question. What are mesosomes? The mesosomes are infolding of the plasma membrane, where they contain certain enzymes which help in cellular respiration. It's not breathing, people, it's respiration, right? So we know what is respiration, breaking down of energy, right? So they help in metabolic processes of bacteria. So that is the significance of mesosomes. So it is marked here. You can see what are mesosomes, okay? So you have to remember this, okay? So usually when we talk about mesosomes, it includes in several ways. They can appear as vesicles. Vesicles are round, okay, membraneless structures, or they can appear as tubules, which are linear tube-like structures, or you can appear like lamella, okay, which are like highly convoluted branch structures, okay, something like branches. So these are called as lamella, okay. So they appear in several forms within the inner foldings of plasma membrane, and that is the significance of mesosome. Now coming to the next one, chromatophores. As the name suggests, we have chromo. Chromo means what? Okay, so chromo means color, okay. Chromo means color. I'm sorry, I was just trying to concentrate on the chat box. Okay, I was waiting for the suggestion for the ink change. So I'll definitely fix it in the next session if we cannot do it now. Okay. Anyways, guys, I'm sorry about the ink. Okay, we'll fix it in the next class. Fine. Functions of the chromatophores. As I was saying, chromo means color, and chromophores, okay, are the bodies that contain color, colored pigments, I can just call it as. So usually, for example, I will give you a name of a pigment, that is bacteriochlorophyll. Okay, bacteriochlorophyll is a type of a pigment. Okay, it is a photosynthetic pigment that are present within the bacterial cell. So usually these chromatophores are present, okay, usually in the membrane enfolding, and they have these photosynthetic pigments, okay, or color bearing pigments that helps in the capturing of certain wavelengths of light. Okay, one of the best examples that you can look about is cyanobacteria, which have bacteriochlorophyll and chlorophyll pigments, and chlorophyll A, which is similar to that of your higher plants. Okay, so you can see here the some kind of vesicular structures that are present along the cell membrane, and this we call it as chromatophores. Okay, so we talked about what are mesosomes and what are chromatophores, and next we will talk about is the nucleus of the prokaryotic cell. As I said, the nucleus of a prokaryotic cell is highly primitive. Okay, so they do not have any membranes. So that means they are non-membranous. So what is the significance of having a membrane, people? Why do we even need a membrane? So just because there is a wall around this, for example, if I have a cell like this, okay, if I have a cell like this, and this is my genetic material, and just because there is a membrane here, does it make the cell very advanced? So what is the more, what is high in this, or what is best in this, and what is not best in here? So what happens is, when you don't protect your genetic material properly, and when you don't have the mechanism to control your genetics, right, control the expression of the genes or the outside attacks on your genes, then you're prone to, okay, numerous changes can happen to you throughout the lifespan, and that can result in abrupt changes in your gene. You can see that most of these bacterial cells, they undergo severe mutations, that is sudden changes in their genetics, where they cannot continue, okay, the proper genetic characteristics for a very, very long time. Okay, that is the first thing. And the second thing is the genetic expression, or the way the cells control, is very abrupt. So cells cannot behave like that in such complex organisms. So now what happened as the cells evolved? So they started putting this membranes around this very important organelles that try to traffic and control, okay, the harm as well as the expressions or the, whatever the functions that they do within the particular cell. So when we talk about prokaryotic cell, they are non-membranous cells. So when inside within the cytoplasm, you can see that you have circular genomic DNA, okay, or circular double-stranded DNA, you can just call it as, okay. So it is a we can't call it as a chromosome. Usually chromosome are like this, right? So we just call it we just call the entire genome of your prokaryotes as a single chromosome or a genetic material. So they are composed of RNAs, they're composed of DNA as well as proteins. All of them are mixed. Okay, all of them are quite completely mixed within the nuclear region. Okay, so apart from the nuclear region, what I told before, that outside the nucleus you can find there is extra chromosomal DNA also present. Okay, so this extra chromosomal DNA that is present, we call it as plasmid DNA. I told you. So this plasmid DNA that is present outside the nucleus is double stranded. Okay, what is this plasmid? Plasmid is double stranded. Okay, circular. You have to remember the features, people. Okay, you will get practice of this because we'll study a lot about this plasmid DNA even in your 12th standard. So anyways, no problem. So plasmid DNA means what is double stranded? DNA will have two chains. Okay, so and they will be in the circular form. So that is why we call it as double stranded circular DNA. All right, so why do we consider this plasmid DNA so much in prokaryotic cell? So you might have heard about antibiotics, right? So we use antibiotics for what? For example, if I fall, if I have a stab, or if I have an infection of any bacterial disease. So when I go to the hospital, the doctor gives me antibiotics. So antibiotics are certain substances. Anti means no, against, okay? Against. Biota means life, against life. So against life means it's basically toxic, right? So when I use antibiotics on bacteria, the bacteria is supposed to die. But because of the ability of this plasmid DNA, okay, so the plasmid will have genetic material, right? So they will try to build a character where even on exposure, like even when you put antibiotics on this bacteria, they will be able to resist. Okay, just like, for example, if you are given snake poison, like for example cobra snake, we know very much, don't do this, okay, usually in many of the yogic cultures they do take a very minute amount of cobra venom, and as they keep on taking for several years, okay, like for example 14 years or 15 years, they develop this resistance towards venom. Natural resistance will build up. Similarly, even in bacteria you can see that this natural resistance to antibiotics, usually that are supposed to kill them. So the, what doesn't kill you makes you stronger. That's what the plasmid DNA does to this bacterial cell. So this is about the nucleoid region. Okay, why we call it as nucleoid region? Because we cannot differentiate between nucleoplasm, okay, and the nucleus. Both are mixed up without the cell membrane. That is why we call it as nucleoid region. And this is about the genetic material of a prokaryotic cell. Now coming to the motile organs of the prokaryotic cell, so we have flagella. So flagella is a whip-like structure. So usually it is like a long tail that is coming from outside the cell. Okay. So if you look at the structure of flagella, we can basically divide this flagella into three significant regions. Okay. So one of the regions that are buried within the cell wall, you can see here. Okay. This regions which is embedded within the cell wall. So you can see here, outer membrane, inner membrane, okay, and the plasma membrane. So you see it is completely buried. So when it is completely buried, this structure is called as, you have your basal body. Okay, we call this as basal body. Here it is. So this structure which is within your cell wall and plasma membrane is called as basal body. Just coming outside of your cell wall is your hook. Okay, just it projects outside the cell wall is a hook. Okay, and next you have your filament. Filament is a long whip-like structure or a tail-like structure where they start beating in a circular fashion. Usually we think that the bacteria moves like a snake like this. No, it's not moving like a snake. So basically it swirls like, it keeps on rotating, the entire cell rotates like this, like a spaceship. Okay. So that is how the swirling movement is created by the flagella. Now, how do these flagella generate so much energy and so much capacity to move this bacterial cell? First of all, when we talk about basal body, which is within the cell wall, okay, and then within the plasma membrane, what we can observe is within the cell wall and within the plasma membrane, the basal body is basically supported by specialized protein structures, which we call it as rods, okay, and then we have the rings, okay, which are at the lower portion near the plasma membrane, that is outer ring and inner ring, okay. So initially what happens is the rings and this rod, what happens, they try rotating, okay, to create this movement in a circular fashion. So the creation of this movement in a circular fashion because of this protein core, okay, there is protein core, this is called as protein core, okay. So there is protein core which is helping in the rotation of these rings. So once this protein core starts spinning, now that will generate energy in the hook, and finally it is transferred to the filament and the movement is generated within the flagellum. Okay. So flagellum is not a hollow structure. For example, if I take this flagellum, okay, if I take this flagellum and then if I cut it here and into several sections, okay, I will make it small, like onion sections, and then observe it under electron, okay, microscope. If I okay, SCO, sorry. Oh, give me a second. Okay, my CRO. Okay, sorry. If we observe it under the electron microscope, we see very, very specialized structures within this flagella. What do we see? The entire flagella is covered by plasma membrane, and within the flagella we have, okay, microtubules, okay, which are made up of a special protein called as tubulin. I'll mention it here. Okay, so these tubulin proteins 1, 2, 3, 4, 5, 6, 7, 8, and I'm sorry, I'll just congest it here. Okay, so well, let us erase this. Oh, sorry, let us erase this. Okay, we'll write 2 more rings. Okay, so you can see here the tubulins are tubulin like a tube like structure. Okay, so these are called as microtubules. Okay, microtubules basically are like tubes which are made up of a protein called as tubulin. Okay, so tubulin protein will construct this tube like structure called as microtubes. Okay, microtubes or microtubule. So these microtubules are arranged. Okay, I'll just erase people. The people who told that you cannot see the ink, I'm writing on the black background. So I hope you are able to see right now because it's quite a lot of writing is going on. Okay, so when we talk about microtubule, okay, it is arranged in doublets. What is doublets? So you have one ring here and you have additional ring here. So let us say that one microtubule as A and other one microtubule as B. Okay, so there are 9 doublets, right? So all these 9 doublets are interconnected, okay, by bridges. What are these bridges called as? Okay, these bridges which connect this, okay, which connect these microtubule doublets, okay, are called as Nixon bridges. So what is the meaning? Okay, one doublet is holding on to each other doublet in a circular fashion, as simple as that. Okay, so on the surface, see, for example, if I take this is a microtubule, on the surface of this microtubules, you can see special proteins. Okay, so these proteins we call it as dynein proteins. Okay, dynein proteins are certain proteins which are present in the microtubules. What they do is they generate ATP. Okay, so they provide ATP or they generate ATP for what? Movement, right? For the flagella to move What is needed? Energy is needed. So these dynein arms that are present on the microtubules, like lollipops, okay? So basically what they do is they provide the energy for this flagellum to move. Now this is about these 9 microtubules and how they're interconnected, and how these candy-like structures called as dynein arms provide the energy for the movement of this flagella. And when you look at the center of the flagella, there are 2 more microtubules. We call it as central tubules, okay, which are interconnected by, okay, intermicrotubular bridge, we can call it as. And then they're surrounded by a sheath called as, I'll write it here, central sheath, okay, central sheath. So the within the central sheath, you have 2 more microtubules, okay. So there are 2 here and there are 9 here. That is why we call this arrangement as 9+2 arrangement, okay. So remember this. And all these microtubules, they start projecting, okay, proteins towards the center, like your cycle spokes, okay, like your bicycle spokes. And these spoke-like structures, we call it as radial, REATs, okay, radial spokes, okay. So radial spokes, what do they do? From outside to, that is, which is in the periphery, no, this is periphery. Periphery means boundary. From outside to center, okay, they project and they hold. Look at this microstructure, which we have to see in the electron microscope, how such complexity in its construction. That is why flagella is so powerful, and it provides tremendous amount of energy, okay. That's what I'm trying to ask. Please change the ink color. So I need a suggestion on how to change the ink color, okay. See, a pointer, I have it, people. So can I use white or something like that? So white or black? White should be good. Okay, I'll try to use white finally. If you cannot see again, please let me know, okay. Fine, people. So we briefly studied the structure of flagella. There is a lot to flagella, so I hope you have understood. So let's move on without wasting the time. So additional structure of the prokaryotic cell that you can observe is your pili and fimbriae. So pili and fimbriae are the structures that arise from the cell wall, okay. So on the surface of the cell, like if you have a cell, bacterial cell, okay, on the surface of the cell wall, you see this hair-like structure, okay. So this pili gets a name because of its protein, okay. The special protein it is made up of is called as pilin, okay. The protein pilin construct this pili and fimbriae structures, okay. So what does this pili do? The pili is basically helping this bacterial cell first in the movement. Second one is attachment, okay. Second one is attachment, okay. And the third one is, okay, sex pili. So what is sex pili? Usually when you see bacteria in unfavorable conditions, like when 2 bacteria are in unfavorable conditions, that means higher temperature, lower temperature, lack of availability of food, or attack of certain invasions or antibiotics, anything you take that can kill them. So during this unfavorable condition, what happens with the bacterial cell is something amazing. So we know that these microorganisms, which are unicellular, they undergo asexual reproduction, right? But sometimes when they have unfavorable conditions, that means the conditions are not good, the 2 cells come close together and they form a bridge, and they connect both of their cytoplasms together and exchange their genetic material, which help them gain new characteristics and help them to avoid from dying or become adaptive to this changing environment. So this is called as conjugation, okay. Conjugation. So since sex pili is responsible for attachment of 2 different cells, and it is also responsible for forming this cytoplasmic bridge between 2 different cells, that is why pili has a high significance with respect to bacterial cells, okay. Is it understood, people? And please let me know if white is okay. Okay, we can still change the ink, now we know that. And the next structure is fimbriae. Now fimbriae is like bristle-like structures, just like your toothbrush, which projects out of the cells, okay. So basically what happens is, whenever you see bacteria in flowing streams where there is movement, continuous movement of water, or the certain bacteria which are highly infectious, so they have to attach the host cells, or they have to attach to this rocky or rocky areas. So this fimbriae helps in attaching to the substratum, like it goes and helps in attachment or anchorage. So this is about fimbriae. So we understood what is pili and what is fimbriae. What is the significance that we have to understand here? Remember here, the protein, pillin, and the role of pili, okay, movement, attachment, and sex pili. And fimbriae, it helps in anchorage, okay, or attachment to host cells or to the rocks in the streams, okay. So what about the ribosomes? So one of the most important cellular organelle that all the cells have is ribosomes. Why? Because all the cells function, okay, from bacteria to us, all of us function in the same way. So how do we function? So we have genetic information in the form of DNA, and DNA, which is present in your nucleus, is converted to RNA or mRNA, we can call it as. And the RNA contains the messages. Okay, for example, if I have been built in a certain fashion, if I have certain character, certain intelligence, okay, certain way of eye color, skin color, hair color, height, you talk about any character or any expression, biological, physiological, cellular, morphological, any of the characteristics you take, it's all information, right? So all of them have to be performed in a specific way. And the cells have to be given the messages to perform such function. All the messages are in DNA, and that DNA messages are decoded into mRNA. And this mRNA, which is carrying the messages, okay, are read by these ribosomes, okay, are read by these ribosomes, okay. Oh, physics, what's the time? Oh my God, okay. I'm extremely sorry, people, okay. We took quite a while to finish this session. So I will try to reschedule so that we will continue from the ribosomes itself. And thank you for reminding about the class, and see you in the next class. Okay. Thank you very much.